Well, I'm going to introduce you to the Hospital Microbiome Project, some of the initial results, the stratagem for actually doing this kind of study, and then Brent's going to take over and talk about the building science aspects of that work. We've got some really exciting preliminary data to show you, and hopefully this can all go ahead and push forward. So for the building scientists among you, we are a microbial ecosystem. We have about 6 pounds of bacteria living on or in our guts and all over our skin. And those bacteria are constantly interchanging and exchanging with the environment we live in, which is changing our perspective upon the initial historical angle towards our approach to microbial life. So, 200 years ago, it would have been very dangerous to have a surgical operation, and now it's really good because we've spent about 200 years trying to kill every single microbe we come across in a way that helps us to eradicate disease. The problem is, it's not quite working as well as it should do. There's still 100,000 Americans dying every year of hospital-acquired infections, and I put that in quotation marks, hospital-associated, healthcare-associated infections in different environments. And even if that's not tragic enough, it's still costing around $45 billion to the US economy, so that should get some interest going into why this might be an important thing to try and eradicate. Now, killing everything in the environment isn't working, and I'll pull from Jessica's book here. We need to think about other strategies. We can't just kill everything. Maybe there are different approaches we need to take. And that's the entire foundation for the Hospital Microbiome Project. It's exploring and understanding the microbiome of this environment, how it moves, what pathways it takes when it transmits around the environment, how quickly those transmissions occur, and what are the other 99.99999% of bacteria in a hospital that are not pathogens doing? And what can we do to take their roles and make them work for us? So the Hospital Microbiome Project is a new initiative. It's funded by APSF, obviously. We had 2 rounds of consultation, setting up a consortium that involved everyone from the architects, building scientists, microbiologists, ecologists, clinicians, the epidemiologists, the lawyers from the hospital, and various other facets of groups to enable us to implement a very sophisticated, I hope, and an integrated study in a new $800 million hospital. And as I'm constantly reminded, your project may be very well funded and you have a large sum of money, but our hospital costs $800 million. And if you say anything to damage the reputation of the hospital, you may be crucified. So on that basis, we're very careful with our language, but we're also not specifically trying to track down and kill all the bacteria in that environment. We're trying to learn how to improve the health of that building and learn to provide a baseline, a knowledge base to enable that. So the hospital started being built about 2 years ago, and we managed to get into that environment before anybody else occupied it. So it opened for business on February 23rd of this year, but we've been sampling in that environment, thanks to APSF, since early January, and we have a daily timescale resolution analysis of air, water, and various other facets. So on the patients, we're looking at nares, hand, and armpit. And the patient room, we're looking at many different surfaces. Some of those are listed here, including the water supply coming into the room, the air supply, and also surfaces of that environment. We're also working with Mark Hernandez and various other people around from the larger consortium to try and investigate the biological particles in the air. We're also doing the staff and nursing stations in those environments. The stratagem is we have 5 rooms and a nursing station on the 10th floor of this building. It's an enormous building. When I initially said, we want to sample this building, it's going to cost around about $100 million. Paula necessarily didn't think that was a good idea. But so we scale back our ambition. We're doing 5 rooms on the 10th floor, which is where you have all of the cancer patients and where you have people who are undergoing transplants. This is high duration occupancy, low patient turnover rooms. And on the 9th floor, where you have people undergoing elective surgery, they generally come in and out during a day or maybe 2 days, sometimes 3. So it's low duration occupancy and high turnover. So that immediate contrast gives us a wonderful hypothesis to test about the impact of duration occupancy and patient turnover, as well as all of the disease states and conditions these people might be undergoing. So Daniel Smith, who's sitting there, hand up, is probably the person who has been doing the most work on this. He actually moved next to the hospital. So he moved house, sold his car, moved next to the hospital in order so he could be there every single day to sample this environment. So kudos to you, Dan. And on that basis, we've managed to get some preliminary data in time for this meeting. Despite the enormity of this project, we've been collecting over 15,000 samples daily over the course of this year. So we're looking at the first 600 samples. We're able to see some interesting data, which Dan was running through last night. So here you see patient room A and patient room B. So room A, I believe, is on the 10th floor, and room B is on the 9th floor?
Other way.
Other way. So yeah. So room A is on the floor which has this high occupancy turnover, and then room B is on the floor which has low occupancy turnover. So people are in their rooms for weeks, if not months. And you see, before the hospital actually opened, you had lots of variability. This is just alpha diversity, the number of bacterial taxa we identified. There's lots of variability there. But then as soon as the hospital opened and all the patients and the doctors and the nurses moved into that environment, the alpha diversity system actually jumped up a notch. So things dramatically changed on the microbial world. And you can also see the little dots, the yellow dots of where cleaning events are taking place in those rooms. And what we see is in certain times a cleaning event is followed by a significant reduction in alpha diversity. Sometimes it's not. Now we know the reason for this is because we get into a room, but we can't always rely on patients leaving a room or dying in a room and the room being cleaned on the schedule which we would like. It happens sporadically, sometimes at 3 a.m. in the morning. So we may take a sample in the morning one day, then someone leaves or dies and is removed and the room's clean, and then we take a sample in the morning and we see an effect of cleaning. Sometimes a patient's already moved into that room again by the time we get to the next sample, and so you do not see an effect of cleaning. And that has a dramatic impact upon our ability to investigate it. In terms of beta diversity, this is kind of how similar the communities are overall in terms of the abundance and well, this is unweighted, but the types of organisms that are there and their relative proportions in the system. When the hospital was closed, both room A and room B on the 9th and 10th floor, they look kind of similar microbiologically speaking. Same types of organisms were there, they were similar environments. As soon as it opened, they both significantly changed in terms of their relationship to each other and the relationship to what happened before. So again, it's pushing that angle that the microbiome is being significantly influenced by the occupancy. And the type of occupancy, either long duration or short duration, is significantly affecting the microbial community structure. We've obviously been working very closely with Jeff Siegel and Brent Stephans and Brent's team at IIT in order to try and implement a really integrated and strategically appropriate building science program. And Brent's going to deal with that in a second. But we wanted to have a look at what impact some of those building science measurements could have upon our ability to interpret the microbial diversity of these environments. So we've got these really cool relative humidity, temperature, and light meters stuck on the rooms of each of the hospital environments. And we see that the communities, the microbial communities on the floor, this is just a tidbit, on the floor of these environments is significantly affected or significantly correlated by the humidity of that environment and the temperature, but not by the light. Now, this goes against some of the hypotheses that I would have come up with, that light's very important in terms of the types of microbes that are in there, but we're still obviously trying to integrate this vast data set. And currently also, this is only the bacterial data. We're also processing these samples for 18S and for ITS, which will get us at some of the protists and fungi that are in that environment. All right, perfect. Dan's been doing some core microbiome analysis to try and understand what microbes may be found only in room A or room B, only in these high occupancy or low occupancy rooms, and what ones are found between them. And when the hospital was closed before it opened, we saw very little core microbiomes in those, very little bacteria that were found in all of those samples. As soon as it opens, we see a massive increase in human-associated bacteria that are found in all of those environments. The humans being in that environment significantly affected the environment, yay. That's a really nice confirmation of our hypothesis. Interestingly, we do find some organisms related to things like Salmonella and Streptococcus infrantis, and we try and keep that quiet from the hospital stuff. This is, here you see people's hands, so the patient's hands in the rooms, and the first sample's obviously taken when we first get to them, but mostly that's just after they've arrived in the hospital. And so you take a sample of their hand then, and we take a sample the next day, and we see usually a significant change in the microbiome of their hands, and in virtually all cases. And this can divide it into the 10th floor long occupancy and 9th floor short occupancy rooms. So the hand microbiota is significantly affected. It's responding to being in this new environment, either because the patient's being treated or because the patient's in a new environment where it's being exposed to different types of microbes. That we have all the data to test that hypothesis. We have all the data to disentangle that relationship, but it's going to take some time and some analysis. Just before I hand over to Brent, so Brent can come up, we've been working with microBEnet, and we just got a news article in Nature today examining this relationship, and we just pull up that. Nature are really excited about this kind of work, and we got this nice thing, a picture of Dan sampling a sink in the bathroom, so that's really cool. And we really are trying to generate a lot of press coverage on this work and get some really interesting data about how the microbiome is affecting these environments out to the public. Now, over to Brent.
All right, thanks, Jack. What do I have, 8, 10 minutes?
Maybe a 2-minute more.
Okay, that's great. So Jack kind of touched on our role. By the way, I'm Brent Stephans. I'm assistant professor in architectural engineering at IIT in Chicago. So we're only about 3 miles or so from the hospital, from the University of Chicago's campus. So we're sort of strategically located there where we can handle this work. So we're working on the building science team. I want to just, we're only about 3 months in. We've got another 9 months or so of data left to collect. So I just want to give you kind of a snapshot of what we're doing in the hospital. And tell you a little bit about our experimental plan, and just show some photos of what we're doing, and a little bit of data just from some random day, just to give you an idea of the data that we're collecting. And this experimental plan was carried out first really by Jeff Siegel at the University of Toronto, over here on the side, and then me and one of my students, Tiffany Ramos, have been working on all of this. So when it comes to the hospital, we're working on the 9th and 10th floors of this $800 million hospital, and then those floors are served by mechanical rooms up on the 11th and 12th floors. Our 2 different floors are served by 2 different large mechanical systems, and these are really huge. I want to show you some photos of these. So sort of the first baseline things from a building science perspective that we want to characterize is what are the ventilation rates? How much fresh air is coming into the rooms on each floor? That's sort of baseline. The building actually tracks this, the physical plant tracks this, but they don't track it very well. We recently sat down with the plant manager and we found all this discrepancy in their giant tracking system. So one of the first questions that arose in our oversight meetings and so forth was, why are you tracking this if the building tracks it? And the answer was, because it's wrong. So sometimes. So the first thing that we're looking at is we're using CO2. These CO2 sensors are about $1,500 a pop, all throughout the building. The easiest thing that we're doing conceptually is measuring the fraction of outdoor air supplied in the mechanical systems for each floor. And this is at 5-minute intervals. So for each data point that we're collecting, we'll have about 100,000 time series data points for each, after the year of sampling, for each type of measurement. So it's a lot of data. And just to give you an idea of the scale of this is me in one of the smaller mechanical systems. These things are the size of this room, some of them. So really monstrous, moving a huge amount of air. So our first question was, can we poke holes in your system so we can measure some of the things that we need to measure? And somehow they actually said, yeah, that's okay. So we've got some equipment set up here. You can see Tiffany downloading data from some of the mechanical systems. So some are way up in the air. We had to get electrical lines run from the plant electrical team. So it's sort of an effort, and it was initiated a little bit late. So we missed out on some of our primary experimental plan because we didn't get approval. We're moving forward with a sort of a secondary experimental plan, and that's okay. So now that we have those at 5-minute intervals for both rooms, or for both floors. Then we have a bunch of ongoing building science measurements in the patient rooms themselves. So this started with some basic sort of baseline supply, return, and exhaust airflow rate characterization. If you look up above you, we were just measuring flow into and out of the room by the mechanical systems. So we did that early stages of the project. From then on, Jack has already touched on temperature, relative humidity, and light intensity. These are really easy, simple measurements to make with these hobo data loggers. Velcroed onto a wall. We've probably got $1,000 worth of Home Depot receipts from implementing this plan, right? That's also at 5-minute intervals. That's also at a nurse station. And these are robust data loggers. Accurate and robust, which are the two things that we're looking for. And somewhat cost effective. That's probably the first thing we're looking for, right? We're trying our best to get human occupancy inside these patient rooms, again, within the budget that we had to work with. So we're doing this with a combination of beam break IR sensors at the doorways. You walk in, it breaks. It doesn't tell you which way you're going. So we can look at that data in a few different ways, and we're still exploring the best ways to look at that. We also have patient room CO2 concentrations. We emit CO2, so the combination of these beam breaks plus CO2 concentrations, both at 5-minute intervals for a year, can give us some idea of how many people are in the room over a certain period of time. And there's actually still some interesting sort of algorithm work that we need to do to couple these sensors together. We're also looking at room pressurization with respect to the hallway. These are all fundamental things that we identified, again, that we can measure robustly, accurately, and cost-effectively that may or may not have something to do with airflow dynamics and microbial succession dynamics at the end of the day. So these are pressure transducers, measuring the pressure difference between inside and outside of these rooms. So far we've seen they're largely at neutral pressure, which doesn't tell us a whole lot. And we're also doing some air sampling via some HVAC filter media. This is probably the trickiest part, but weekly samplers are taken from filter media installed over the return. It's sort of a free pump. The HVAC system that's working is a free pump for us to do sampling. So just to give you an idea of what this looks like in the patient rooms, most of what we have looks like any other medical device, and there's lots of humming and beeping in the environment, so some of our noise constraints actually aren't that big of a deal in this hospital. Here you can see the hobo data loggers just inside the room. Here you can see a pressure sensor. Everything's enclosed in these sort of Radio Shack-like boxes and tacked on the wall. Lots of kind of groundwork on getting the right batteries and so forth so that we could get long-term data logging capabilities without having to go into the patient rooms too often, trying to be as minimally invasive as possible. You can see there's a little clear tube sticking out of the corner. So part of our weekly visits are to maintain that and re-stick that said tube and so forth. So just the nuts and bolts of this are kind of interesting. We've got CO2 sort of tucked in the one place we could actually get power. I think we got approval for this. I don't even know anymore. It's just there.
It's confusing after all.
It's confusing. It's humming along. Every once in a while someone takes a tube off, or, well, we haven't had anyone unplug it, I don't think. Okay, these nuts and bolts are just interesting to deal with. And then you can see on the outside we have these beam break occupancy sensors. We've had actually very few casualties of all this equipment in 3 months. We lost one sensor lately, but a week later it magically reappeared. So we have no casualties, actually, which is not what I expected. So with just a couple of minutes left here, I think I wanted to show a couple of snapshots of data just in the last couple of days. I picked out April 15th, tax day, it was a Monday, just pulled this out, just to give you an idea of what our 5-minute data look like and how we can inform some of the data analysis going forward. So here we have patient room temperature from those little data loggers on the wall for 5 different rooms on the 9th floor. We have code names for these floors. So 101 through 105 with the black, green, blue, red, and orange there. And you can see, this is just a 24-hour period pulled out of our data set. We see oscillation, that's temperature controlled by the HVAC system. And you also see some some dips, right? So this tells us something happened in this room. Someone changed the thermostat, maybe a new occupant. It gives us some evidence. So the one thing that Jack showed earlier were 24-hour average temperatures that we pulled. That's certainly one way to look at the data. Noon to noon, I think, is what Dan was doing from one day to the next to say, well, what was the average temperature? Well, does that capture the full story? I don't really know. It also tells us that you can't necessarily jump in and do a spot measurement of temperature. You need sort of a history. So that's all fair here, I think. same sort of data from the same day for relative humidity, and we could see differences between rooms, but general trends overall. And these could result from basically these rooms are all being served by the same air handling system, so it makes sense that they should track. We've got light intensity, which actually follows a pretty, basically tracks the sun more or less, and or occupancy with artificial lighting. We don't really know this yet. But we again see some variations from one room to the other. Room 103 at that time received a lot more light. At its peak, that received about the same amount of light as a television studio. So very, very bright sort of environment at that time. So how do we look at this data? I don't know the answer yet. How do we handle this data to compare with Jack and Dan's team with their data as we go on with this project? Here's a good example of patient room CO2 in these same rooms on the same 9th floor. Another 24-hour sample, 5-minute intervals here. We see differences in absolute magnitude, say between the orange and red, those couple of rooms relative to the green and blue. They're tight, so they're near instrument uncertainty, and that's another story. But what this does show us is, you know what, there actually might be greater occupancy in those top two rooms because their ventilation rates are the same, more or less, because they're controlled by the same system. What this also shows is that the black line down there reads 0 because, and it's unfortunate that I pulled this data out, but it went in succession. And I think this must have been when someone, well, either a data logger died or someone took a tube out and stuck it in somewhere else, maybe a kid, who knows. So that's field work, right? We've already lost 1 bit out of 15 data points. That's not bad that I've shown you. And then just to finish up, we've got some occupancy data. Again, these are interesting data to sort of deal with. What we're looking at, total beam breaks. So just how many times was this beam broken at a doorway on this 24-hour period of midnight to midnight of April 15th. And what we see is, does this tell us how many people were in the room at that time? No, not really, but it tells us how active the room was, right? And if we look at 104, 105, with higher number of total beam breaks, we actually see higher CO2 concentration. So we're working to try to tie this together to see what we can do. And this is what the actual 5-minute beam break data look like. It's all over the place and sort of hard to process. So with that, I want to thank Dan, and Jack wants to thank Dan, Tiffany, Sloan Foundation, and the Center for Care and Discovery, the hospital, and much hugs and kisses. Yeah.
Well, just to suggest a clarification, you said we emit CO2, people emit CO2. The study isn't emitting CO2.
Yes, we as people, yes. I don't, only when I show up.
Brief comment. All of us are trying to get other people interested in this program through names of pathogens. In our work with water, we become very sensitized to the importance of de-identification of sample size. It's a real problem. Municipalities don't know about this. Moreover, when you mention sort of working mycobacterium and a lot of other things, people get real uptight with what it is. And we don't know what it is. Next gen data sequence is too short. Definition is too imprecise. So we just have to be very careful about this nomenclature. Do not say to the New York Times, look, we found streptococcus this and that. See, it's the most pathogenic organisms we're organisms. Anyway, I just do have to be careful.
On the basis of that, I mean, like that article in Nature that you guys in Nature, we just did, it says, you know, we were very explicit, we didn't find anything in concerns.
That's important.
Sure, yeah. We got in trouble with that with an article on the front cover of the Tribune, where they took what we put on the internet and said, oh, they found these organisms in the hospital. And we were like, oh, well, actually, but they're not dangerous. We have no idea if they're dangerous.